My name is Todd, and I'm joined by my group mates Elena, Simon, and Jimmy today, presenting simulating cell membrane physics and endocytosis process with soft body physics.
Okay, let's talk about soft body first. So soft bodies are basically a bunch of springs connected to each other and into different shapes. By adjusting like the forces between the springs, we can achieve different simulation effects, like for example a piece of clothing or like a jelly, or in this case we simulated a cell membrane. So to simulate a spring, we take reference to the Hooke's law, which states that the force acting on the object hooked to the spring is equals to the spring constant times the displacement between the object and the spring's equilibrium position. About the simulation time step, it's basically the same as what we did in homework 4, but it actually allows our springs to stretch infinitely and resulting a loosely connected membrane and allowing objects to fall through it to break through the membrane directly. To solve this problem, we apply the XPBD model, which is to apply different constraints to our soft body simulation. This time we applied like a distance constraint on each spring, and if we detect that the particles are overshooting, we just calculate the correct position and move the particles to the correct position to correct them. However, this also extends into another problem, which is that as we are connecting particles with each other, the force acting on each particles are actually depending on each other. So the problem is that if we run the time step sequentially, we could end up being still overshoot despite running the constraint on our springs. We solve this problem by splitting single time step into X substeps and repeatedly calculate and correct the position of each particle to achieve a better effect in simulating physics and soft body physics in general. This is very simple in terms of coding. We just add a for loop here and then run the time step calculation multiple times with the splitted up delta time. Finally, we add damping to our model, which results in damped oscillations in each of our spring. This simulates drags as it slows down the oscillation a long time, and this can simulate our desire to like simulate the cell membrane inside a water environment.
Now, let's talk about possible applications of soft body physics. Simulations enable us to create, observe, and study systems that would be otherwise hard to explore. A good example is studying a cell membrane. Cell membranes are highly complex structures made up of phospholipid bilayer that gives a soft and flexible structure like the movement of amoeba. Take this double-layered cell membrane model for instance. We have used spears of yellow and orange to symbolize individual lipid molecules. These spears are interconnected like a mesh, and each joint behaves like springs. Their dynamic interaction lead the membrane to its fluidity and flexibility. This enables the membrane to deform and stretch like clothes under collision, mimicking the nature of a real-life cell membrane. We can add more features to our membrane to mimic other cellular activities. For instance, we can simulate endocytosis, the process by which cells engulf and internalize biomolecules like this.
We can also simulate a cell and how the nucleus within the cell can control its movement. As you can see, the cell didn't collapse into a layer because we've applied internal pressure to each of the vertex, so it could maintain its shape even without the support of the nucleus. And you can see how it's following the movement of the nucleus. We've also implemented a very simple interactive controller for us to test out different settings of membrane. Like such, just click on the screen and it will spawn some particles to collide with the membrane. So that's our presentation. Thank you for listening. This is Group D.